Good morning, good afternoon, and good evening. Thank you very much for joining Stream B, the Conservation Genomics session. My name is Rebecca Johnson. I'm the Chief Scientist at the Smithsonian's National Museum of Natural History, and I'm your chair for the next hour. We have a packed lineup. It was a really brilliant lot of submissions that we had for conservation genomics. So in the interests of balancing taxonomic diversity, technique diversity, and of course our speaker diversity and career level, we have a whirlwind of talks for you for the next hour. We've got 2 keynotes that we're going to hear from. We've then got 5 short presentations following that, and then we will try and share the questions equally among our speakers. So I'm going to dive right into it and now hand over to Jamie Lethwaite, who will be our first speaker for today. Thanks very much.
So I want to first thank my collaborators and co-authors, Arna and Phil, who are the engines behind this work. And secondly, to dig into this main question of my talk, I wanted to start off with an example that's relevant in my neck of the woods, the Pacific Northwest. So many populations of Pacific salmon in this area are in decline, as you can see from this figure, showing commercial catch numbers for 5 species, which are virtually all trending downwards. And so there's huge interest in protecting and recovering these populations. However, a huge challenge lies in the fact that within these species, there are many genetically, ecologically, and spatially distinct population groups. Above, you can see the map of just one species, the chum salmon, and its 9 distinct population groups. On top of that, existing conservation budgets in Canada are not nearly enough to manage all the threats and conserve all of these populations. And so we're faced with a tough decision. If you can't save them all, how do you decide which ones to prioritize? So often higher priority is given to unique populations, but the criteria can vary widely as what you define a group as unique. You can use geographical boundaries, morphology, or ecology, but obviously these criteria can be qualitative or imprecise. In salmon sister species, the rainbow trouts, researchers decided to use genetic distinctiveness to rank populations from most distinctive to least distinctive, where more genetically distinct populations have higher conservation priority, and I've highlighted those with the red stars there. Now, using genetic distinctiveness as a prioritization tool is becoming more and more common, especially as sequencing gets cheaper and cheaper. And this study used all genetic data. They did not distinguish whether this distinctiveness was due to adaptive variation or neutral processes. Now, why does this matter? Well, distinctive populations may arise simply as a result of drift, and they may actually carry high loads of deleterious mutations, especially in small populations. Or within their population, they may have reduced genetic diversity overall compared to other populations. In that case, prioritizing distinct populations might actually be counterproductive to the conservation of the species as a whole. And so instead, some authors have argued that we shouldn't incorporate information on adaptive variation when prioritizing populations. One potential problem is that identifying which subsets of the genome are adaptive is hard. It takes time, it takes money, it takes effort. There's a question of which traits do you measure, and it's increasingly looking like most traits are the product of many loci of small effect, which makes the detection of outliers and that adaptive variation very hard. And so the question arises, do we even need to do this? What if you could get similar results by skipping that step and just using overall variation? And so we performed a simple test. We prioritized populations based on overall genetic variation, and then we prioritized populations using only genetic variation that was identified as adaptive. And then we asked if similar populations are prioritized based on the 2 strategies. The first step, of course, is identifying that adaptive variation. To do this, we use data from 2 very different species, the yellow warbler and lodgepole pines. To identify adaptive variation, we used 2 very different approaches. In warblers, we used a genotype by environment association, where we extracted climatic variables for each population and asked which SNPs are most highly associated with climatic variation, which was interpreted as evidence of local adaptation. In pines, we used a genotype-phenotype association. So the original authors had already identified and measured 4 fitness-related traits, And so we used this data to identify loci that were associated with adaptive phenotypes. For each population, we separated out that adaptive genetic variation from the overall genetic variation, and we performed a PCA on each set of SNPs to measure genetic differentiation. Each shape here represents a different population. Then to represent that genetic differentiation, we took the centroid from the PCA and we built a pairwise distance matrix between each population. We transformed this into a neighbor net, a phylogenetic network, and we calculated Shapley values, which is a measure of evolutionary distinctiveness. Finally, we gave a ranking based on that distinctiveness to each population, and then we compared rankings from both overall and then adaptive variation. I'm going to start with the results from the warblers. The populations are listed here, and then the first column is the overall variation, and the second column is adaptive only. Populations are color-coded and numbered according to their genetic distinctiveness, with red and lower numbers being higher ranked and green being lower ranked. And so we see a very high correlation in rankings between the two approaches, with a correlation coefficient of 0.76. And highlighted in the red boxes here, you can see that 4 of the 5 top ranked populations in both sets are the same. When you look at the spatial distributions of these prioritizations, where left is genome-wide and the right is adaptive only, we find that peripheral populations, the ones on the edges, are often more genetically distinct when compared to central populations in both adaptive and overall variation. Moving on to the pines, again, we find that rank distinctiveness scores were highly correlated to those resulting from all SNPs, with a rank correlation of 0.74, and this time 3 of the 5 top ranked populations are the same for the 2 sets of markers. Again, populations of pines near the edges of the range tended to have higher priority in the red, and so higher distinctiveness in both sets. So overall, we found that we get very similar results when you rank populations using overall variation and then only the adaptive subset of that variation. This means that we may not actually have to do that additional step of identifying adaptive variation when we are ranking populations for prioritization. Of course, we've developed this general framework to approach this question, but we've only applied it to 2 species so far. It needs to be tested on many others. So if you have the appropriate kind of data set for this question, please contact me. I would love to test it out on your study species. Thank you.
Hello, my name is Brad Schaefer, and I'm here to give you a brief update on a very ambitious project we've been working on for the last year or two called the California Conservation Genomics Project. As the title suggests, this is a project that seeks to work with agency and resource managers to use the power of landscape genomics to better inform and enable effective conservation in California. Now, for those of you who may not be aware of it, California is the 3rd biggest state in the United States after Alaska and Texas. It is the most biodiverse. 1 in 8 people live in the state, and it has the most endangered and threatened species of any state in the country. For all of those reasons, California is both an incredible hotspot for biodiversity and conservation, and also an incredible challenge for effective conservation and management. Now, if you, like me, were interested in doing conservation genomics work in the state of California, for example, on this beautiful spadefoot toad, a species we work on a lot, you might do something like this. The first thing one would do is go out and collect spadefoots from all across the range in the state of California. We'd collect some genomic level data, some SNP data set from RADseq or target capture, perhaps even whole genome resequencing. We would then analyze those data to get a sense of the scope and scale of geographic variation, perhaps using a principal components analysis. We might dial in a little more and use a Bayesian clustering analysis to look at finer scale resolution. And at the end of that time, what we would have is a sense of genetic variation across the landscape for one species where that species occurs. But what if our goals are more grandiose than that? What if rather than one species, we want to in fact characterize genomic variation at the landscape level across the state of California. We want to do it for plants and animals, for marine, freshwater, and terrestrial taxa, for vertebrates and invertebrates, all across the whole state, and generate maps and resources of genomic variation that cover the full breadth and width of the biodiversity of the state. That's the California Conservation Genomics Project in a nutshell. The CCGP at a glance, right now we have identified and funded work on 224 species across the state of California. We are in the process of generating reference genomes for about 125 of those species. And for each species, we will generate not just a reference genome, but also a full set of resequenced individuals, 150 per species across the state, which should result in somewhere between 18 and 20,000 resequenced genomes. Some of the features of the program, we're developing a dedicated core facility so that all extractions and library preparations will be done in a similar way. We're funding uniform genetic coverage, whole genome resequencing only at about 10x for each individual, and developing common pipelines both on the bioinformatics side and also on the landscape genetics side. So how are we doing with this project? Well, here's a map of California that shows the USDA different ecoregions across the state in black and some of our major groups. And I'd say we're doing pretty well. These are just individuals across species for marine taxa, for terrestrial plants, terrestrial vertebrates, and even the more difficult to work on terrestrial invertebrates, mostly insects. Taken together, we have the range of the state really well covered at this point. What will we do with those data? Well, I can't show you because we don't have the data in hand yet, but here's an example from a much more modest RADSeq data set that we completed for the foothill yellow-legged frog. And these are the kinds of interpolated genetic variation maps that we hope to create for each species using not just RAD data, but whole genome resequencing. What's more, we can take these individual species maps, which are valuable in their own right, stack them up, look for patterns across those species maps, ask whether regions are consistently areas of high genetic variation that might be resilient to climate change, ask whether those most important regions are connected via protected corridors, and if not, how can we protect them? And similar kinds of analyses. Our progress to date is pretty good. The funds have been distributed, the species and localities have been determined. Most of the samples, although not all, are in hand. We have about 20 reference genomes that are in play, including a few that are now on the sequencers. Our goal is to have most of the reference genomes and something around half of the resequencing done by July 1st of 2021, and all the data completed by July of 2022. That's the goals, that's the hopes. Before I sign off and take questions, I just want to thank funding from the state of California. This came directly from the state legislature, also Illumina. Our collaborators include 70 PIs from all 10 UC campuses, a variety of different state and federal agencies, and the UCLA LaCrette Center for California Conservation Science. Okay, thank you very much, and I will look to take questions.
Hi everyone, my name is Rémi Alliot. I'm a PhD student from Montpellier in France. I'm working in phylogenomics and comparative genomics, and today I would like to show you how sequencing genomes from rat kill sample can be useful in conservation. In Africa, several species have different distribution, with a population living in South Africa and a second population living in East Africa. In our study, we decided to measure the genetic distances between the two populations of aardwolf and butchered fox that you can see here. Indeed, for both species, two subspecies have been defined years ago based on their distribution and their morphology. However, no genetic studies have been conducted yet to characterize this population. In a conservation matter, it's important to know if the two populations are species or just subspecies. First of all, by measuring the genetic distances observed between our pair of population using two mitochondrial markers, we found that the genetic distances observed between the two populations of aardwolf were relatively high and might be considered as interspecific genetic distances. To confirm this result, we decided to look at the genetic distances of this population at the genomic scale. We thus decided to generate two hybrid reference genomes using both Oxford nanopore long-read sequencing and Illumina short-read sequencing. To do so, we first developed an optimized protocol to sequence mammalian roadkill tissues with Oxford nanopore technologies. Using this protocol, we were very glad to obtain two high-quality genomes for both species. Indeed, with about 10,000 counting and an N50 value of about 1 megabase, these two genomes rank among the best carnivoran genome in terms of both contiguity and gene completeness. Then, to estimate the genetic distances observed between the southern and the eastern population of our two species, we sequenced one additional individual from South Africa and one additional individual from East Africa. Then, we defined a genetic differentiation index which, like the FST statistic, is an index of genetic structuration. However, this new index is based on the heterozygosity of several independent genetic regions. By classifying the heterozygous sites in 4 different categories, such as the heterozygous sites private to the population A or to the population B, we are able to have an estimate of the population structure. For the population A, we used 2 individuals of this population, and for a global estimate, we used 1 individual of each population. As for the FST statistic, the more the 2 populations are structured, the more the genetic differentiation index is close to 1. As you can see in this graph, when we measure the genetic differentiation index for the southern population, we do not have any structure. The index is close to 0. However, when we measure the genetic differentiation index for the species, that means with the 2 populations, we observe some structuration. Because it's difficult to define a threshold to consider subspecies as different species, we created a dataset composed of 4 additional pairs of well-defined species of carnivores. For those species, we used the same number of individuals and we estimated the 2 same indexes. These additional pairs of species allowed us to determine a threshold between intraspecific and interspecific genetic distances. As you can see, the 2 populations of aardwolf are more structured than the 2 well-defined species Canis lupus and Canis aureus. Given this result, we think that the 2 species of aardwolf might be considered as different species. Regarding the badged fox, even if the structuration observed for the 2 populations is higher than the structuration found within all pair of species, it is more difficult to conclude. So as general take-home messages, we can conclude that roadkill samples are underexploited resources for genome-wide analysis. The genetic differentiation index can be estimated with only 3 genomes and might be used for species delimitation analysis. And finally, southern and eastern aardwolf may be considered as distinct species. To finish, I would like to thank my team in Montpellier and my collaborators for participating in this study, the committee of the conference Biodiversity Genomics for giving me the opportunity to present this work today, and thank you for your attention.
I'd like to start by thanking the organizers for inviting our talk. Today we're going to be talking about field genomics and biodiversity documentation in Madagascar. So as most of you know, Madagascar is one of the Earth's hottest biodiversity hotspots with 80 to 90% species endemism across all groups, and the endemism is severely threatened by human impacts. The endemism and the biodiversity is extraordinary, and there's high levels of microendemism, and it would be tragic to lose this biodiversity, and certainly we want to understand as much about it as we can. Very tragically, however, the Madagascar is very poor, and the Malagasy people are forced to encroach on the forest simply to meet their daily needs, and this is causing habitat destruction throughout the island, and this endemism is thus highly threatened. So we report our first efforts to deploy field genomic techniques to understand and document the biodiversity of Madagascar. The publication came out this summer in Conservation Genetics, and I invite you to have a look at next generation technologies applied to age-old challenges in Madagascar. We report the field deployment of nanopore sequencing technology and mini-PCR in order to understand and document Madagascar's biodiversity. We have several goals. First, simply to document the biodiversity using a DNA barcoding approach. Secondly, and going forward, to deploy whole genome resequencing for a population genomics approach. And finally, and most importantly, to transfer this knowledge to Malagasy scientists and students. The DNA barcoding approach is particularly important for these cryptic radiations. This is my personal favorite, the mouse lemurs. So we are looking at 6 species here, which to our eyes look virtually identical, but they're genetically quite diverged. So we need to use a DNA barcoding approach. We're using cytochrome B and placing it in a phylogenetic context simply to understand which species we're looking at. Now going forward, we want to take a population genomics approach using whole genome resequencing. We're starting with an advantage in that we have a beautiful near chromosome level reference genome. I invite you to look at the paper that was published in 2017, Larson et al. In BMC Biology. We used multiple platforms to get to this near chromosome level, and we were even able to characterize the centromeres. So I think you will find it of interest. Now we're going to deploy this whole genome resequencing where we can use medium coverage Illumina sequencing to see what's going on the ground. This is a view of forest in southeastern Madagascar, which looks pretty homogeneous. But if you look very carefully, you can see that there are gallery forest habitats near this small river, and the rest of the forest is dry. My colleague, Jörg Ganshorn, who has been working in this region for a very long time, reports that we see one species that is only found in the dry forest and another in this gallery forest. But as I like to say, these animals are certainly within sniffing distance. And in fact, Jörg was certain, based on microsatellite data and other sources of evidence, that there were hybrids. We have now used RADseq data to look at these hybrids and have discovered that in fact they're pure parental species. So we're quite interested to know what is going on at the genomic level in this contact zone. And so that is a next step for us. Our work in Madagascar is much facilitated by the lab that Phoebe and Liz have set up, Mahaliana, so that we really can take things to a next level with regard to genomics. And finally, and importantly, Lantu, who is here in the foreground, Lydia and Marina, have hosted a series of workshops for Malagasy students, and they've been well received, and the students are very excited to use these mobile genomic technologies in their own research. So I end by thanking you for your attention and acknowledging all of our many collaborators and funding sources, and thank you again for your attention.
Okay, hello all. I recently completed my PhD at the University of Sydney and the Australian Museum, and I'll be discussing a couple of chapters from my thesis. So the red-tailed black cockatoo, or Calyptorchus banksii, is clearly a stunning bird, as you can see here. The males on the right with the redder feathers, and the females on the left with the more orangey yellow feathers. It's found in eucalypt woodland throughout Australia, and it's characterized in 5 subspecies based on morphology. And so very little genetic information is available for this species, even though it's a iconic Australian species as conservation concerned. So there are a lot of unanswered questions on its evolution phylogeography. And you can see the samples we obtained from the birds here. The circles are the frozen tissue samples and the stars are the topad samples. And I'll talk to you a bit about that now. So we obtained samples from 4 museum collections throughout Australia, 113 frozen tissues and 47 topads, some very old topad samples, over 100 years old. And basically these are just a thin slice of topad off a taxidermy bird. You can see here in the bottom right, and we extract DNA from that. And I used DartSeq, which is a ddRAD type platform, to generate SNPs in these samples, and did some mitochondrial DNA sequencing. And 62 of the topad samples worked, which was reasonable. A lot of the fails were replicates, so that was all right. And we called over 24,000 SNPs when combining this whole data set. We really wanted to do a thorough quality control because these topads likely contain a lot of degraded and low template DNA. And we found there was higher error rates in the topads when using replicates compared to, say, the frozen tissue samples. So we wanted to know how this might bias some of our inferences. So we performed a number of analysis and titrated through some SNP filters and see how it affected our error. And so one bias was that older samples tended to have more false homozygotes. So heterozygotes erroneously called as homozygotes. So there's sort of a temporal bias in diversity. Older samples seemed less diverse. So we had to take that into account. And then we performed various population genetic and phylogenetic analysis. So I'll show a few results now. So based on various SNP analysis, we found 5 distinct genetic clusters in this data set. And this is an example of a structure plot based on SNPs. And as you can see, the 2 northern species, this purple cluster, there was no differentiation between them. In the Sammuli inland subspecies, there was clearly 2 distinct units here. And then the naso and graptogyn subspecies really stuck out. The mitochondrial DNA results were a little incongruent to this. So these 3 subspecies sort of stuck out still, but then the northern haplotypes were scattered throughout this network, and some of the central eastern haplotypes were nested within the northern. So we wanted to use this SNP data to characterize evolutionary significant units and conservation units. This is traditionally based on reciprocal monophyly of mitochondrial DNA, but this can be problematic, especially when there is mitonuclear discordance, and we don't want to base these inferences on just one or a few markers. So we analyze these SNPs in a phylogenetic framework using SNAP, which considers the coalescence of each SNP marker. And we compared a number of conflicting groupings in this phylogenetic framework. So these groupings are based on our morphology or their previous taxonomy and some of the genetic results we got. We found that these 5 groups that we saw in this cluster plot was the most highly supported phylogenetic model. And so traditionally when we used to characterize ESUs, we would treat them as separate units and keep them separate, and they were treated as separate entities. But now a new paradigm shift is when characterizing these conservation units is identifying at-risk populations that might be suffering from genetic erosion, and then figuring out strategies on how to mitigate this genetic erosion. So we also provided estimates of genetic diversity and interconnectedness to manage the genetic diversity of this species in a broad evolutionary context. So to wrap it up, so this is the subspecies before we started the study, and this is the subspecies now. So Macarinchus and Banksii was synonymized as Banksii. And these Western Australian Sammuli populations were actually more closely related to that Southwest naso subspecies than the rest of Sammuli. So we named this new subspecies Calyptorhynchus banksii esconditus and assigned it this lovely looking holotype. So these subspecies is sort of how we characterize ESUs, so they were sort of synonymous in this sense. We found that this graptogyne subspecies was likely suffering from genetic erosion. It had the lowest genetic diversity, it was the most divergent, and there's only about 1,000 individuals left. So we suggest strategies on how to augment this genetic erosion. And so we suggest genetic rescue strategies, but instead of using the highest pairwise kinship, which you would normally do, we suggest using the lowest pairwise kinship subspecies to augment this population, which would be Samueli, because these subspecies are so divergent and there is a risk of outbreeding depression. So thank you to everyone here, particularly my primary supervisors, Nathan Lowe and Rebecca Johnson, and thank you for listening.
So first of all, I'd like to thank Rebecca Johnson and the organizing committee for inviting me to give this talk about our research on biome shifts in Africa. So the study we're presenting today is entitled Diversification in the Savannah Species Tetraploid Rainforest Radiation in the African tree genus Absilia. The rainforest is the oldest major vegetation type. It was the most common biome in between 56 and 33 million years ago, but it has been largely affected by the global cooling and acidification of the climate. And this actually caused large-scale rainforest extinctions in the rainforest lineages about 33 million years ago. In contrast, the savanna is a much younger biome. It began to expand around 60 million years ago, and it became a well-established biome very much more recently, about 8 million years ago in the late Miocene. This plot illustrates this global cooling and acidification of the climate between 55 million years ago and today that we were mentioning, with a minimum in temperature and humidity at 33 million years ago, which caused all these large-scale extinctions in the rainforest. And if we look at the more recent periods, basically the Miocene, Pliocene and Pleistocene, we observe very significant climatic oscillations of the climate. These climatic oscillations caused several cycles of expansions and contractions of the rainforest that really influenced the evolution of the biomes and led to biome shifts. To illustrate this, that would be the African rainforest covered today. But if we go back 8,000 years to the humid period of the Holocene, we observe that the two forest blocks in West and Central Africa were actually forested. They were connected by rainforest, so there was a forest expansion. And we go back 20,000 years ago to the last glacial maximum, we observe that the rainforest was very fragmented in favor of the expansion of the savanna biome. So this contraction as expansion cycles, as I said, probably led to many biome shifts in many tropical rainforest lineages in Africa, but actually these African lineages have been very little investigated. So in our study, we investigate the tropical legume tree Absilia. Absilia is an important timber tree in Africa, so its populations, natural populations, are threatened. And it has currently 7 species, 2 savanna species, north and south of the equator, respectively, and 4 rainforest species. There is a 7th taxa, which is little documented, which is Armeria peturae. For our studies, we have been using molecular markers to investigate the evolution of the biome shift in the genus Absilia in Africa. In our earlier studies, we were using single sequencing, and for our more recent studies, we're using genome-wide data. Our results clearly show that the rainforest and the savanna species fall in different clades. In addition, these clades have different ploidy levels. The savanna species are diploid and the rainforest species are tetraploid. This result clearly indicates that there's been a single biome shift in the evolution of Absilia in Africa. And interestingly, if we date our species trees, we observe that the savanna clade diversified earlier, about 12 million years ago, than the rainforest clade, which started to diversify for 4.2 million years ago. This finding of older diversification of the savanna clade versus the rainforest clade is pretty unique, as in most other lineages, mostly in the Neotropics and a few that have been investigated in Africa, show an older diversification of the rainforest, as this agrees with the fact that it's an older biome. However, our data didn't allow us to conclude on the direction of the biome if it happened from the rainforest, although the dating suggests that it could be from the rainforest into the savanna. The topology of our tree doesn't resolve really the relationship between these two clades, the savanna and the rainforest clade, and the sister genus Insia. Therefore, the direction cannot be solved, and we would need to sample all the outgrowths, the sister genus Insia and the genus Brodigracia, which are in the same clade as Auxilia, to resolve the direction of this biome shift. In addition, for the savanna clade, we perform a population genetic study, also using classic markers, nuclear markers at least in our earlier studies, and then updated with wide genome data. And interestingly, we found that for the two species, we find very little genetic structure, which is not geographically, which is not correlated with the geography. This clearly indicates that there's been no genetic signal of fragmentation of the savanna, which probably suggests that the savanna during these climatic oscillations were able to migrate north and south and were not fragmented, as it was the case of many rainforest lineages, which were fragmented during the coldest and most arid periods of the Pleistocene. And just to finalize, I would like to thank you, my co-authors. Miriam Hurt was the co-led this study with me. She's based at the Institute of Agricultural Research in France, in Bordeaux. And Armelle Dompegan was working on this project as a postdoc, and is now currently, she's an expert in systematics of Auxilia. He did his PhD in this topic, and now he's based at the INRA of Toulouse. And also I would like to thank many people who are involved in the project, either people who are providing lab assistance or fieldwork assistance or discussion about the results that we obtained. Thank you.
I'd like to start by thanking the organizers and my co-authors. And Michelle, George, Anders, and Tom, who as postdocs, did most of the work that I'll be presenting. Today I want to illustrate ideas about the conservation of adaptive potential using 3 of our study species to provide examples of key points. First is the orange roughy, a long-lived, slow-breeding fish inhabiting the deep sea at about 700 to 1300 meters and breeding on banks and seamounts, and note Faraday seamount at the middle of the figure there. Based on 14 microsatellite loci, we found no significant structure within the North Atlantic among these marked locations. We only found differentiation between the North and South Atlantic with an FST of about 0.015. However, genomic data based on about 4,000 SNPs showed some North Atlantic structure at neutral loci, shown on the left side of the figure for both FCA and DAPC ordination analyses. And on the right side of the figure, you can see that most of that structure collapses for putative loci under selection. And the selected loci are based on FDIST, PCAdapt, Bayes scan, and Bayes ENV2, with only Faraday seamount samples standing out. The second species, the short-bearded Grenadier, lives mostly in the abyss, and we focused for this species on sites either side of the Mid-Atlantic Ridge and north and south of the subpolar front. We found no structure at 17 microsatellite loci. However, genomic data showed some small differentiation either side of the ridge for neutral loci, and again, clear differentiation at putative loci under selection for regions south of the subpolar front. Here, SE1 and SE2, and red and green dots in the figure. Although we have some preliminary data on possible relevant loci, the key point is that across an environmental boundary representing a transition for both sea temperature and salinity, there's the potential for adaptive population structure not revealed in neutral loci and suggested by the data for both of these species. Final species I want to talk about today is the round-nosed Grenadier, which lives over a remarkable range of depths. And so here we focus on differentiation in the vertical dimension of the sea. We sampled in the same location off the Hebrides on the same day and sequenced nuclear genomes for 14 to 16 fish from each location for a total of 60 genomes. Making Manhattan plot comparisons across the genome with habitat depth as the trait factor, we found little effect for most comparisons, but a strong set of outliers for all comparisons against the samples from 1,800 meters, shown at the bottom of the figure here. At 6 functional loci, we found non-synonymous changes that were fixed at 1,800 meters, and there was no differentiation by depth at about 5 million putatively neutral SNP loci. An important feature of this system is that the young remain at about 1,000 meters and segregate by depth as adults. This inference is based on extensive catch per unit effort data shown in this latent class model. The segregation is according to genotypes at the loci we identified, and there is also some morphological differentiation in adults at different depths, such as the differentiation of gape size, shown here to the right of the figure. While I don't have time to discuss possible functional differences at the identified genes, adaptive variation across this point of transition is perhaps not unexpected, since it reflects a number of changes, including the loss of ambient light, thermal and saline transitions, and the oxygen minimum layer. So the key points are that evidently adaptive diversity can vary in both horizontal and vertical dimensions in the deep sea, distinct from patterns at neutral loci, and therefore not addressed by management strategies that are based on population structure assessed only at neutral loci. And with that, I'd like to thank our sponsors, a list of people too many to read out, who helped with the project, and you for listening to me Zoom through this talk.
Hi, my name is Erin Wilder, and I'm a researcher at the San Diego Zoo Institute for Conservation Research. And today I'm going to talk about genomics of fitness-related traits and chromosomal variation in the endangered Pacific pocket mouse. So some outstanding questions in conservation genomics include, what are the underlying causes and predictors of inbreeding and outbreeding depression, and how can we manage genetic diversity and maintain fitness of small populations? The Pacific pocket mouse is a really great system to investigate some of these questions. It was historically distributed across the coast of Southern California and was known to a number of localities up until the 1930s. And after that, it disappeared and was actually thought to be extinct for several decades until it was rediscovered in these 3 populations in the 1990s and emergency listed under the US Endangered Species Act. Investigations of genetic variability at microsatellite and mitochondrial loci in the 3 populations, Dana Point, South San Mateo, and Santa Margarita on the left, showed little contemporary connectivity, and notably only a single mitochondrial haplotype in the Dana Point population was found after 2008. Effective population sizes were small in all 3 populations, but especially in Dana Point, where NE was around 2.3. Continued population declines prompted a conservation breeding program at the San Diego Zoo, where 10 to 15 founders from each population were brought into the zoo, were brought in to found the conservation breeding program. An admixture over the subsequent 8 years led to population growth in the program. But despite this growth, it was clear early on that not all populations had even fitness in the breeding program. And individuals from Dana Point had fewer offspring, leading to a decrease in ancestry of Dana Point in the breeding program. So we looked to genomics to help us understand the underlying source of these fitness differences. We first upgraded the reference genome by combining HiFi reads and OmniSeed data to generate a new reference genome that was orders of magnitude higher than the old reference and much more complete. We also generated low coverage whole genome sequence data for hundreds of individuals from the wild and from the conservation breeding program, numbers that are unusual for an endangered species and that were enabled by the conservation breeding program. So one of the first things that we were able to do was with the more complete and more contiguous genome was to estimate long runs of homozygosity, which are indicative of recent inbreeding, and more accurately estimate inbreeding coefficients in the population. So here on the left, I'm plotting individual inbreeding coefficients for each of the 3 wild populations and the captive breeding program. And you can see Dana Point with the largest inbreeding coefficients. And on the right, I'm plotting runs of homozygosity across the largest 85 megabase scaffold. And you can see one of the Dana Point founders shows these large runs of homozygosity compared to a Santa Margarita founder. And then an F1 hybrid between the 2 populations shows virtually no runs of homozygosity, as you'd expect. We can also look at changes in runs of homozygosity over time, and notably when we look at the Dana Point population at inbreeding coefficients, we can see an increase in runs of homozygosity starting in 2008. And this is consistent with what we know from census data, but here we can see it in real time using the genomics. A mitogenome was not previously available for this species, so Caitlin Curry from our group was able to pull the mitochondrial reads out of the PacBio data and assemble one de novo. And we can use this to help us understand the evolutionary history of the maternal lineage in this species. And we can also use the more contiguous genome to help us impute genotypes that we can use to correlate with fitness traits that have been collected in the breeding program, like adult weight and body condition, survivorship, mate choice, litter size, and so on. And Anne Mizuraka and Marlis Hauk from the San Diego Zoo have been karyotyping PPM for years, and they have seen that Dana Point individuals have a different chromosome number than the other populations. But with the karyotyping, it's so far been unclear if these differences are fixed. So we're excited about working with DNA Zoo to generate some high-speed data to add to this data set and try and shed light on some of the chromosomal variability and whether it's fixed between populations, which might lead to outbreeding depression. So with that, I'd like to acknowledge my funding and take any questions.
Huge thank you to our speakers in the Conservation Genomics session. We've heard everything from mega diverse ecosystems to species delineation. We've heard about making the most of what you've got by scraping things off the side of the road, and what is the adaptive potential and how does it relate to conservation. I'm going to spin around and trying to get a question to every one of our speakers. And firstly, a huge thank you. There's lots of appreciation in the chat function to say thank you to you all. My first question is to Jamie. Why is genetic distinctiveness crucial? Is an overall amount of adaptive genetic variation more relevant as a predictor for evolutionary rescue? And great talk.
Great question. So I would counter by saying that indeed genetic distinctiveness is already being used by conservation practitioners to identify populations for conservation prioritization. But they're not, these studies rarely identify which subset of that genetic distinctiveness is adaptive. So my question was, do we need to do that? The literature suggests that we do, but I want to actually pursue that question. It may be that overall genetic distinctiveness captures that adaptive subset, or it may be that, so in my 2 species, there was high gene flow between those populations. It may be in species where there's more fragmented populations, more isolated populations with less gene flow. You may see contradicting results between adaptive and genetic variation when ranking populations. So I've only tested this in 2 species, obviously needs to be tested in many, many more. I'm just proposing a framework for actually answering which is more important or if adaptive information is actually important when prioritizing populations.
Thanks so much, Jamie. It's such an interesting aspect that we're going into in conservation genomics, the idea of adaptive potential. Really great questions. Next question is for Brad. Any tips for developing similar projects in other regions, and what has been the most challenging aspect of this project so far?
Yeah, can you hear me? Yes, I think so. Great. Applying it to other areas and what have been the most challenging. And in many ways, they're sort of two sides of the same coin. Interestingly enough for us here in California, so this project, we got about $12 million from the state of California. That was a line item, a budget item in our 2019 state budget. And getting the funding, which is what everybody always says is the hardest part. In this case, it just was a matter of reaching out to the right people and talking with the state legislature to get that funding in place. That was actually less hard. And maybe it's because we knew the right people and we had some wonderful political allies here in the state. The hardest part, and I think the biggest challenge anywhere, is getting 70 PIs from 10 University of California campuses who care about, nothing but runs of homozygosity and deleterious mutations, or care about nothing but systematics, or nothing but file geography, whatever it might be. Getting them all to pull in the same direction so that we would have the kind of uniform, consistent sampling, the kind of uniform, consistent genomic coverage, where we could combine all of those data without having our colleagues who are real analysis wonks getting upset because you're trying to combine apples and oranges and that never works. And so of course we can't get anywhere. And so that has been without a doubt the biggest challenge. And probably a secondary one is saying of the, I don't remember what it is, but many thousands of species in California, how do you pick 200 or 300 of them? And how do you have a process which is open and transparent and completely above boards and competitive, and yet fulfill a certain set of goals and missions? It's taken us the first year of the project to accomplish that, but we've done that, and I think we've done it well and fairly. So I think that we will see in the next 2 years as the data roll in and we develop these consistent pipelines for consistent analyses. But I think we're hoping it will serve as a model for other parts of the US and for other parts of the world, because I think there's tremendous value in it. Okay,
thanks so much, Brad. Well done you on balancing those very different stakeholder needs and securing the funding. Our next question is to Remy. Great talk. How fresh were the roadkill, and how did you have to modify DNA extraction and prep to compensate?
Yeah, thank you. Well, some tissues were fresher than others, but for MinION sequences, we chose the freshest one. Then, to extract the best part of the tissues, we found that conserving tissues in RNA later was better than in alcohol. Then it was important to eliminate the dead cell for the extraction. And finally, we use a particular ratio of impure beads to extract the longest fragment of DNA of the extraction. And after a few months of development, it works.
Thank you, Remy. I think there's a lot of interest in the protocols, in many of the protocols that have been shared here today. So I encourage everyone to reach out to our speakers either via Slack, or many of them are on Twitter, and I'm sure that they'll be happy to share more. Thanks again. Now, our next question is for Anne. Why are you using the cytochrome B gene and not cytochrome oxidase 1 as a barcode? Thank you. Very interesting talk.
That's a great question, and I, with apologies to Rebecca. I'm going to try to answer both questions, if I may. The cytochrome B question is very easy to answer. It's historical precedent. I started sequencing cytochrome B when I was a graduate student, and so we just have an enormous database of cytochrome B sequences, and it's very reliable, so we can trust the primers, and it's just very non-glitchy, so that it's just purely technical, really. And the mitochondrial locus, as you know, is a single non-recombining locus, so we would get the same results. But going to, there was a question about how what we're doing is going to actually protect the forest, connect the dots there. And that is a beautiful question. It's a question that we ask ourselves all the time as conservation geneticists, genomicists. How is our empirical data actually going to have an impact on conservation practice? And it's a complicated answer, but the short answer, I think, is our outreach to the Malagasy community of scientists, that we are transferring knowledge in such a way that they will have they are the stakeholders, and it will allow them to conduct the research, invest in their own natural resources, and therefore have the incentive to work with local people to try to stop the progressive deforestation. It's very complicated, but that's my best in a short time frame. So thank you for that question, for both questions.
Thank you so much, Anne, much appreciated. Next question is for Kyle. Great work based on the museum samples. Did you consider the use of date/year of collection as one of the parameters to see shifts of genetic diversity in time?
Thanks, Rebecca. Yeah, very good question. The red-tailed black cockatoos are super long-lived species, really long generation time, probably about 14 years. So we didn't really expect much of a shift in genetic diversity over this sampling period. Plus, some of those really old samples, there was the problem with the bias in diversity due to the false homozygotes. So it's difficult in this system, but we did do this exact thing for another species, the Lord Howe Island woodhen. So we analyzed old topaz samples before a bottleneck and compared them to modern samples and detected and quantified the genetic bottleneck. And that was recently published in Animal Conservation, run by Richard Major and Mark Eldridge. So yeah, it's definitely possible, but maybe not for the cockatoos system.
Thanks, Kyle. Definitely a challenge with archival specimens, but not insurmountable. Very impressive. Rosalia, our next question is for you. Great talk, Rosalia. Very impressive work. Do you see your findings informing current day management?
Thank you, Rebecca, for your question. This question will mostly apply to the savanna species, which are the most used for their timber. We were very surprised to find that there are not really any genetic signal of loss of genetic diversity, despite the heavy use of this tree. In the population genetic study, we found a clear pattern of isolation by distance, which indicates limited seed dispersal of this species, which really discards a huge human intervention in terms of exchange of genetic material across regions. We suggest that the local populations use the local genetic resources. And we also found a clear, for both savanna species, we found a clear signal of genetic connectivity over time and over large distances, which suggests that these species can migrate responding to human pressure and climate change.
That's really impressive. Thank you for that. Now, our next question goes to Russ. This is such an economically important species. Thoughts on how this could relate to management?
I'll be brief. The orange ruffy is indeed an economically important species. In fact, it was quickly extirpated in places where it was hunted. A lot of these species are taken as bycatch, but increasingly as more accessible fish become overexploited, people are turning to the deep sea. And so it's important to consider the deep sea. And I guess my short point about management is that a lot of the diversity regionally in the oceans is difficult to assess and cryptic. and especially for these broadly distributed marine species. So these genomic methods allow us to identify potentially cryptic differentiation, especially the adaptive variation, I think is potentially very important. And that's really what the talk was about.
Thank you. Now we've got 20 seconds left, and there's actually been quite a few great questions for Aaron. I encourage you to reach out to Aaron via social media or via Slack. And again, I'd like to thank all of our speakers for some wonderful talks today and a snapshot into why genomics is so important for conservation. Thanks again for joining us.